Hi, and welcome to the AP Biology Lab 5 Cell Respiration podcast. This podcast I'm going to talk about the worm lab or the pea lab or the pea and the worm lab. But before we do that, I should talk a little bit about respiration. Respiration occurs in mitochondria, and it occurs, remember, not only in animals but in plants as well. And it even occurs to a degree across the membranes of bacteria. And so what is the function of cellular respiration? It's to break down organic compounds in the presence of oxygen to release ATP. And so this is kind of a cool diagram. It shows all of the different creatures of life. So we've got plants, algae, and bacteria. We've got us down here with the other heterotrophs. And essentially what you do in cellular respiration is you're taking the energy found within those organic compounds and in the presence of oxygen converting that into water, carbon dioxide, and then ATP. Now the interesting thing about plants and other autotrophs is that they're able to convert that carbon dioxide, water, and energy from the sun back into those organic compounds. So what are the specifics of cellular respiration? Remember, there are 3 steps. There is that glycolysis, where we break that sugar, which is a 6-carbon molecule, down into 2 3-carbon pyruvates. Then within the mitochondria itself, we break that into acetyl-CoA, and eventually in the Krebs cycle or the citric acid cycle, we turn that carbon dioxide out, and we make a bunch of NAD and FAD. And that NAD and FAD in the electron transport chain make a whole bunch of ATP. So this is the overall equation. Glucose plus oxygen yields carbon dioxide, water, and then a little bit of energy. And so in this lab, what are we measuring? We're actually measuring the oxygen that's being consumed. And so in respiration, we're using something called a respirometer. So a respirometer looks like this. It's just a glass tube. It's got a weight on the bottom of it. So the first thing you need to put in is a little bit of cotton. This is absorbent cotton. So you put that in the bottom. Then you put a little bit of KOH, and I'll talk about that in a second. And it's kind of nasty, so I'm not going to do that right now. And then you put some non-absorbent cotton over the top of that. Then you put your organism that you're going to study in here. And so if we were to do, for example, germinating peas, we'd measure out a certain amount of germinating peas, and they'd go in here, or we'd put a worm in here. And then we want to make sure in all our other respirometers that we control the volume, because volume is going to be really important. And so we usually use glass beads are added to the respirometer, and that makes sure the volume in each one is exactly the same. And then we stopper it, make sure it's really airtight at the top, and now we've got a respirometer. And so you've got your organism in here that's going to respire. You've got this pipette that goes out the side, and then what you'll do is you'll just set it in water. And so in this diagram right here, it shows that. So we're putting all of our respirometers right here in water, and then we just let them sit and equilibrate. And so in other words, the temperature inside there has to be equal to the temperature of the water. We then put them underwater, and then we're going to watch the water bubble move in. Now let's go through some of the important things that I just mentioned. First of all, KOH, or potassium hydroxide. As you're respiring, in other words, as you're taking in oxygen, you're also giving off an equal amount of carbon dioxide. And so the volume inside the respirometer would never change if we didn't have potassium hydroxide. So the function of potassium hydroxide is it actually grabs onto the carbon dioxide and turns it into a solid. And that solid inside the respirometer has very little, if not a negligible amount of volume. And so what that does is get rid of that volume in the carbon dioxide so we don't have to worry about it. What else is added? We've got beads that are added to keep the volume, remember, exactly the same in each of those different test cases. And then finally we let it equilibrate so the temperature is in the same. And so you can use any respiring organism, be it a pea or an earthworm or corn or whatever. The tricky thing in this lab is actually reading the pipettes, because they're going to be submerged underwater and the numbers on them are really, really small. And so we should take a second to look at those. On this diagram right here, this would actually be the water that's flowing into the respirometer in the blue, and then this white area is going to be the air bubble, and that air bubble extends all the way into the respirometer. And so right here we can read the meniscus at the bottom. So if this is 0 and this is 0.1 milliliters, it would actually be at this point at 0.07 milliliters. And then as it flows in, we could read it up here, and it looks like it's at 0.22 milliliters. And so we could find the difference between that over a given period of time, and it's going to tell us how much of the oxygen was actually consumed. If we look at some analysis for just a second, the analysis on this gets a little messy. First of all, you'll have beads in one of the respirometers by themselves, and the function of that is to just control for changes in the temperature. So there's going to be fluctuations within that water during that period of time, and so we'll actually subtract that from our values to find the difference, and that's going to tell us how much of the change in the volume is actually due to respiration. Next, how do we plot this data? Well, we plot it like this. So we're going to put time across the bottom, and then on the side we're going to have the milliliters of oxygen consumed. And so in this data set right here, you can see that the germinating corn at 22 degrees, or the warmer, is going to have a higher respiration rate. And as I say that, the slope of this line, if you ever have gathering data on the y-axis, and then you have time on the x-axis, the slope of that is going to give us the rate. And so if we look at these, we can calculate the slope of each of those and then represent that over here in a bar graph. And so now we have the different slopes of each of those, and so we can figure out which one of those had the highest rate of respiration. And so that's the cellular respiration lab, and I hope that's